to introduce the first speaker. Hopefully he's backstage somewhere because I haven't clocked him in a little while. He's there? I got the thumbs up. All right. I think he might need a very warm welcome. Yes, this is Steve.
Hi everyone.
Hi Steve.
Hi Stevie. Hey, look, I'm going to be honest. I'm not really that comfortable with this talk. Ebony asked me 6 months ago to do this talk, and I started preparing the slides on Monday. When I showed them to her, she said, no, these slides are not appropriate for Nerd Night. So there's a little bit of science, but I'm going to try and break it up and sort of give you a little bit of a story. But as you can see, I'm going to be talking about wastewater, which everybody, well, no one in this room probably thinks about wastewater, but everybody should, because you've all contributed to it today. So I'll start off. So I'm a research and teaching academic, and I'm a molecular microbiologist. And this photo here is my current lab. So I have 2 honours students and 6 PhD students who are currently working in my lab. And we work on mobile genetic elements and study how bacteria move from one cell to another. And essentially that research is kind of telling us that bacteria are becoming resilient and resistant to all different types of antimicrobials. But what we try to also do is try and determine ways to eliminate these problem bacteria to try and control them as an alternative to antibiotics. So that brings me to the type of mobile genetic element I'm going to talk to you about today, and that is bacteriophages, or for short we call them phages. So what they are is bacterial viruses. This is not what they look like, but every time I'm putting a cartoon. But it kind of has, yeah, you'll see some real pictures of bacteriophages throughout this talk. So bacteriophages are viruses that infect bacteria. And they have these different types of, there's two types of life cycles. So this here is a bacterial cell. And when a bacteriophage recognises its receptor site on the bacterial cell, it can undergo 2 different pathways of replication. So there's one where the genetic material of the bacteriophage integrates in the chromosome and remains dormant, and these bacteria then continue to replicate, and each daughter cell receives a copy of the bacteriophage in the chromosome. But the type of, the life cycle I'm going to talk to you about today is the lytic cycle. So some bacteriophages are strictly lytic. So once they inject their genetic material, it takes about 20 minutes for for the bacterial cell to replicate multiple phages and kill that bacterial cell, and those bacteriophages are released and can go ahead and infect other bacterial cells. So the good thing about these bacteriophages is that they are self-dosing. So if their host is present, they continue to replicate. And it's a great alternative to an antibiotic because these bacteriophages don't care whether the bacteria is resistant to an antibiotic or not. So it will kill it either way. So that's my introduction to these viruses or these bacteriophages. So now I'm going to give you a little bit of an introduction to wastewater treatment plants. So this is what a good wastewater treatment plant looks like. These wastewater treatment plants operate worldwide. So it's been over 100 years that they've been using this system called the activated sludge system, where it's a robust system for treating wastewater, and it contains fecal material and undigested food remnants. So you can see that all our wastewater is collected in these basins and it's treated, but it's treated with bacteria. And bacteria, the whole purpose of treating wastewater is to remove excess nutrients. So this simple picture here just shows you where we've got the influent there, and that's where all our waste enters this aeration basin. And they're collected, and all the waste is collected, and then there's various different processes that occur where these basins are generally aerated, so there's bubbles in there. And the reason for this, and they're kept there, that waste can actually be treated or be kept in the aeration basin for over a week. And the whole purpose of that is to enrich for bacteria that will remove phosphorus, nitrogen, carbon, all these compounds, because what then happens, it goes into this secondary clarifier, and the wastewater, well, the activated sludge will settle, and the clear water at the top can then be reused. And most often it's actually it actually gets into another water body, like in oceans, lakes, rivers. Sometimes they use some of it for irrigation purposes. And there's even been talk about using it to drink, but when we've had droughts. But because it's actually clean, it's so well cleaned. But if there's excess nutrients in there and that gets released in another water body, we end up with algal blooms. So it's very important that this process is operating properly. So this is what a healthy activated sludge aeration basin looks like. And because these are microbes that are essentially removing all these nutrients, this is a biological system. So biological systems can fail, like any biological system. And I'm going to say something because my PhD student Jessica really wants me to say this, but these systems can also get sick, like a person and any other biological, an animal. And what I'm going to talk to you about now is the type of one way that these systems can get sick. And what it is this wastewater foam. It's not the wastewater, it's not a foam that you generate when you're washing your dishes with the morning fresh. That's an unstable foam. These foams are very, very stable, and you can see these are all in Victoria. All these photos are taken in Victoria. And you can see some of them are just covered constantly in foam, some of them overflow, some of them cover walkways. And these foams are so thick and viscous that if you took a sample and put it in the fridge, which we do in the lab, not at home, those foams will remain stable for 2 years and they don't break down. So when this happens at a wastewater treatment plant, they need to fix that straight away, because if the general public are still going to the toilet and still flushing things down the sink. So that needs to get fixed, and it's costing the industry, it's estimated to cost the industry billions of dollars. And guess who pays for that? You guys, in your water bills. So when you get your water bill, it tells you how much you've got, how much water you've used, and it's often quite little, and then you've got all these costs associated with it. And some of that will go towards the wastewater treatment plant for it to continue operating. So now I'm going to tell you, so there's been heaps of research in why this foam is so stable, viscous, and very difficult to break down. It's caused by gas bubbles, and we can't do anything about that with wastewater treatment plants because the aeration process encourages the bacteria to grow that remove the excess nutrients. There's surfactants. So surfactants are detergents that we put down the sink, or some bacteria can actually make really potent surfactants. But what stabilises these foams are these hydrophobic particles, and in this instance it's bacteria, and I've written down here they're filamentous bacteria, and the most common, so 90%, and this doesn't only occur in Australia, it happens worldwide. 90% of foam are due to this organism known as Gordonia amare, and they'll become clearer in a minute. So this is what Gordonia amare looks like under the microscope. It's got these right-angled branching kind of filaments, and this stain here is actually a foam sample. And if you look at it, it looks like a pure culture of the bacterial cell that on its own. And what we also know is that related organisms, I've written down here, Gordonia, Rhodococcus, Supermarella, and other Mycobacterium, some people may have heard of that. These organisms all have a large amount of mycolic acid in their cell wall, and this mycolic acid makes these bacteria hydrophobic. So they've been given a name known as the mycolata. So the mycolata are responsible for foam stabilisation, Gordonia amare being the most common foam, most commonly found in foams. So there's absolutely no reliable methods to treat these or to break down these stable phones. So what a lot of wastewater treatment plant operators do is they use some of their wastewater that they've treated and they spray it on top. That can help bring down the foam a little bit. Others throw in chlorine. And when you throw in chlorine, that's going to kill the good bacteria that's removing all the nutrients, so we can cause other problems. So there's no reliable method. So I introduced you to bacteriophages earlier, and now I'm going to tell you what I in 2008. So in 2008, I was employed as a postdoc and I moved to Bendigo for 3 years. And here's me when I moved to Bendigo. And the project was, it was an ARC linkage grant together with Melbourne Water and South East Water. And the idea behind it was to isolate as many bacteriophages for these microlata strains and to develop a cocktail that we could then reapply to the wastewater treatment plant. So isolate the bacteriophages from the wastewater, grow them up in the lab, and then reapply them and see if they would kill the bacteria that's stabilising these foams. Now, this list, I just want you to see that this is the list that I got given when I started my postdoc, and they said all these organisms have been isolated in foam, have been isolated from stable foams. So start screening for bacteriophages for them. Gordonia amare being the one that they really wanted to target the most. So we started to screen for phages, and these are what the bacteriophages look like under the electron microscope. So these are real bacteriophages that we've isolated, and as we were isolating these, we were sequencing and characterising these bacteriophages to make sure that they're safe to be used. And we've now got a collection of over 100 different bacteriophages that can be used as a cocktail to treat these wastewater to use in a wastewater treatment plant. We've used lab scale experiments where we've tried to use these bacteriophages in a cocktail and see if we can reduce cell numbers and even break down foams that we generated in the lab, and they look like they're working really, really good. And we also found a lot of novel phages. We isolated a bacteriophage, the smallest bacteriophage to be isolated to date. We found a lot of novel findings throughout our screening process. But what was... so I mentioned different genera of bacteria, and Gordonia is one of them. And Gordonia amare is the species that we really want to target. So we isolated many different phages for Gordonia species, and these are all our phages, and we've published them, we've published their genomes, we've tested them against all of our Gordonia strains. They're able to lyse or kill all of our Gordonia strains except for Gordonia amare. And this becomes a problem because Gordonia amare, more than 90% of the foams are stabilised by this organism. And so then I mentioned we started this project in 2008. In 2015, I joined Trobe, so I left the university system altogether, but then I came back in 2015. And there was some email exchanges with phage hunters in the USA, the Science Education Alliance in the USA as well, asking us if we've isolated a bacteriophage for Gordonia amare. So everyone around the world is now screening for bacteriophages for Gordonia amare with no success, which is really unusual. Sometimes it's a bit difficult to isolate a bacteriophage for certain organisms, but not this extreme. So what we use, Gordonia terrae, which is a related organism, has had over 2,000 bacteriophages isolated and sequenced at the time. Gordonia amare, none. So this was really unusual, and we didn't know why. And at the time, the Gordonia terrae genome was sequenced, and they couldn't see anything special. But there was a comment made saying that it didn't really have any antiviral systems. So then in 2017, I actually got funding for a project to genetically modify bacteriophages, and I employed a postdoc, Dr. Stephen Martinovic, who's sitting at the back over there, Give everyone away to Stephen. Anyway, so Stephen joined my lab and he was working on a different project, but Stephen now works for Nanopore, and he loves sequencing. He's very good at DNA sequencing. If you need anything sequenced, you can go and speak to him and he can help you. But at the time, I took out the Gordonia amaranth strains that we had, and we had the type strain, which was isolated in the USA probably 40 years ago. Then we had one from Germany, and we had 4 strains from Melbourne. So Stephen sequenced these genomes, and when he sequenced them, what he found was that they actually encoded. Firstly, he was able to divide them into 2 different species because their genomes were different. So we introduced Gordonia pseudoamare and Gordonia amare. So both of these organisms are responsible for foam stabilisation, but they contained an abundant amount of antiviral systems. So these bacteria had evolved in a way that even if a bacteriophage had injected their genetic material in there, the cell would be able to destroy the bacteriophage. So it was able to protect itself from these bacteriophages. So at the time, we thought, well, no one's ever going to isolate a bacteriophage for Gordonia amare, so let's just, we'll think about writing this up and publishing it. And then in 2019, I got this honours student, Jason Rose, who's also sitting at the back. Now, Jason was a very interesting person. His project was to actually use our bacteriophages that we isolated and see how long they'd survive in wastewater if their host wasn't present, if their host was present. But as a side project with every honours student, I normally give them a little side project, and if it doesn't work, it doesn't work. If it works, it's great. And Jason's side project was you have to screen wastewater treatment plants for Gordonia amare phages, knowing very well that no one around the world can isolate one. So Jason, so now I want to tell you a little bit about Jason. So this is where I want to, so about Jason, not a morning person. See this photo here? This is from Nerd Night. This photo here, I told him to prepare himself for the photo. Can you tell that he just got out of bed? Not a morning person. So at the start, when he started honours, the first thing that we taught Jason to do is try and come into uni before 1 p.m. Maybe we can get an experiment done. He's not a day person. He's also not a day person. So at 4 o'clock in the afternoon, he'll often say to everyone in the lab, I'm struggling today. But right now, he's wide awake. At night, he's wide awake. So he's not a morning person, not a day person, struggles to stay awake. And he turned around and asked me today for this talk, because did they ask you to talk shit at Nerd Night? Because we're talking about wastewater. So that sums up Jason's personality. I want you to know that Jason finished his honours, masters, and is about to hand in his PhD as well. Okay, so Jason, well, because he was coming into uni after 1, he couldn't really start his honours project, but he thought he'll screen for a phage. And a few months into honours, I see Jason and I'm like, Jason, he's like, oh, you know that thing you told me to do? And I'm like, yeah. He goes, well, isolated it. And I'm thinking, so I've got Stephen telling me he's sequenced all these genomes. There's no way we're going to isolate a phage for Gordonia amare. Jason, who's half asleep all the time, telling me that he's isolated a phage but hasn't started his science project. And other people around the world emailing me saying, what's going on? Has anyone isolated a bacteriophage for Gordonia amare? No one can do it, but the guy that's half asleep in the lab is telling me. So, Jason apparently isolated a phage for Gordonia amare. Nobody believes it. So anyway, there was a lot of discussions between Jason, Stephen, myself, and there were repeated discussions, and a we found it hard to believe. So I first thought that everything was contaminated that it was working with, and told him you have to prove that it's the right thing. And what we have here at the bottom is a lawn plate. So this is the bacteria Gordonia amare growing, and as we're diluting it down, you can see single plaques. That's what we see with a bacteriophage on a plate. So we sequence this. We extract the genetic material and we sequence this organism, and it turns out it's not a bacteriophage at all, but it was the first environmental Sakari bacteria species. Now, what this is, when next-gen sequencing came out, everybody started sequencing all these different environments, and we could sequence everything, and they referred to this as the microbial or biological dark matter, because nobody can grow it and nobody knows what this does. It's found in wastewater, in the oral microbiome, it's found in other environments, it's found in your stomach, hence why it's in the wastewater treatment plant. So the wastewater treatment plant is like an external gut, if you want to think about it. Most people think about it as an environmental source, but it's really just an external gut. We then grew this we grew this organism up, and we tested its ability to kill the other mycolata, and it was able to kill most of them. So all of a sudden we had something that was able to kill Gordonia amare and Pseudoamare, plus all those other mycolata, and that's because of Jason, who isolated it.
I think this test is fine.
Of these antiviral systems,
whenever we have it more dynamic, as we walk from one side of the stage to the other, they will appear off screen, like triangle, she went all the way like gone.
Gordonia amare, despite the fact that it actually can
this won't happen.
All these antiviral systems.
So I wouldn't be concerned. This is mostly fine.
So we have to name it.
Yeah, you can have a look here.
So we named it Microbacter analyticus. This is what it looks like. So
they all
very
need to sit
small and has all these
between
organelles, and we don't know what any
within
of them
that black box. If it goes out, that's bad.
We named it Myco because mycolata related organisms.
It's fine.
For Trobe bacteria because it can only grow with Gordonia amare, and Liticus for lysozymes, for the ability to kill. But in our lab we call it JR1, Jason Rose 1. It's actually named after Jason. It's JR1. So you can come to my lab and say JR1, we all know what you're talking about. So we looked at it under the electron microscope even further, and you can see here these filamentous bacteria are the host bacteria, the Gordonia amare, and all those all these little cells here are the parasitic bacteria. So when you mix them together, you can see they all attach to the bacterial cell immediately. They're like magnets. After about 4 hours, the filamentous bacteria are gone, and it's just them present. So we've done, I'm not going to go into details on the studies that we've done, but we've done a lot of studies on these parasitic bacteria, and we now know they will only grow in the presence of their host, and we know that it essentially eats them up. And these are SEM, these are scanning electron microscope pictures. I don't know if you can see them clearly from the audience, but they're like a 3D image, and you can see all these little cells attaching to the filaments. And so this was quite exciting because we were looking for a bacteriophage, and Jason isolated something that was really, really unique. So then I asked him, he needs to tell me how he isolated this bacteriophage. And so we go to the wastewater treatment plant, we collect the samples. He put a photo here, not wearing a lab gown, so I'll talk to him about that later. He's in the lab getting the wastewater. And he filters it through like we're looking for a virus. And then what normal people do who work with bacteriophages is this is a lawn plate, this is the bacteria, and he's spotted that onto this. And you can see all the bacteria growing. Because he's can't get up in the morning and all that, if you leave it a little bit longer, so normal people will stop here and will throw it out. We'll throw that plate out, say I've got nothing, throw it out, that's it, we're done. But But because he would have left he would have left these plates in for weeks probably, and he would have seen that, and so that's how. So what we worked out is that this parasitic bacterium actually eats its host, and you need to incubate it for a longer period of time for it to get to the point where you've got clearing, like what we're seeing here. So I just wanted to show this video because I love this video, but we then we collaborate with people at Bio21 and they do electron tomography. This organism here at the bottom is our host bacterium, the Gordonia amare, and this little thing here is the parasitic bacterium. You can see that as soon as you mix them together, they attach, and we wanted to see what structures we could see, because we don't know anything about these organisms. Now these guys, the microscopic people, they're like, oh, can you see that? Can you see? We can't see anything. So we have to sit there and just say, yeah. But then they did this for us, and you'll see this in a second. Because, I mean, unless you're a microscopist, I don't know how they see these things, but they coloured them for us. And they did this. And you can see these, there's a tube that pierces the host, there's some pili, those purple pili attaching to the host, and so we still don't know how it does the killing, but we do know how it does the infection. And that's because, and we also don't know what many of those structures are. So what we're now doing, what Jason did during his PhD is he was able to, so say this is Gordonia amare, our bacterial cell. We know that there's mycolic acid in the cell wall. Jason had generated several mutants that were no longer able to make mycolic acid. And those mutants that were unable to make mycolic acid are no longer being able to be killed by the parasite. So the parasite attaches to the mycolic acid. The mycolic acid is what makes them hydrophobic, which causes foaming. So if we applied and used this parasitic organism and put it back into wastewater treatment plants at high concentrations, chances are it's going to kill all those, it'll eliminate most of those bacteria that have the mycolic acid in their cell wall and that are responsible for foaming. So, to sum up this talk, in our lab we've now isolated greater than 100 different microbiologicals. Gordonia amare and Gordonia pseudoamare have been sequenced and characterised. Mycobacter analyticus, or JR1, has been isolated, sequenced and characterised. JR1 demonstrates a broad lytic activity, so it can kill many different mycoloidal hosts. And our findings indicate that JR1 requires mycolic acid for host attachment. That was Jason's PhD that he just published this year. And we're now collaborating with researchers to identify and characterise the cell surfaces, so all those little structures that you saw in the images, so that we can understand and try and understand better how this infection process occurs. And we're looking at using JR1 as a potential control method to control wastewater foaming, which will reduce everyone's water bills and make everyone happy. So we're now also exploring JR1's role in the human gut. We know that it's present in the gut at high levels. And we're collaborating with people at the Hudson, where we're looking at the presence, and then we're going to be looking at, we're also going to be looking at the organism's interaction with human hosts. So studies are in progress to evaluate JR1's therapeutic potential against bacterial infections. This image here, Jason told me to put it in because he thought it would be funny. So there's a Pac-Man in. So with that, I just want to thank my lab and all the people involved in this work.
I'll tell you what.
I can get that.
You can take a breath now, Steve.
Excellent.
So, for those that don't know Steve in the audience, usually have to kick him off the stage, so I feel like he deserves an extra round of applause for actually sticking mostly to that. Questions. So I might get some house lights tests. Does anyone have any questions for Steph? For Steve. I've misgendered you, Steve. Sorry.
It's gone room temperature.
Yeah, it's fine.
The battery went out.
Yeah. I'm virus free now.
I don't drink from that, so.
Yeah.
So I'll repeat the question.
Jason's hilarious.
Yeah, so if there is a foaming event, is it more about prevention or treatment?
That's a really good question. Prevention is going to be the best option, but in terms of applying, so we've thought about this a little bit. So most of those extreme foaming events that I showed you where the foam was overflowing, they generally occur when a wastewater treatment plant is new and just starting. And,
let me try this one.
but the thing is, it's going to be like if we could use it for to actually fix the problem once it's occurred, that would be great. But it would have to be more of a used more for preventative method. I think it's, they're already found. These parasites are already found in wastewater treatment plants, and like I don't have any evidence for what I'm about to say, but maybe we'll do a project in a few years. But I think it must be an imbalance. So if the parasite is there, they never get foaming problems, and if it's there at a certain abundance. But if they are not there, or if they're depleted for whatever reason, that's when I think foaming becomes a common problem.
It's like 26.
Alright, any other questions? Yes.
Yeah.
You could just use Parmesan all the time.
No, I think Parmesan comes with chips.
No. Great question.
Repeat the question.
Alright, so Lauren asked, is JL1?
Is Jason in your stomach? That's a bit questionable.
Everything else was on there.
So JR1 is the parasite, but the Lugonia aberrant has only ever been found in wastewater treatment plants.
Curious if they've changed.
It's not found.
Everything I said is the lowest.
The parasite, we don't know. So why the parasite's there, we don't know. However, some research conducted on the oral microbiome, they've isolated a relative of JR1
At the beginning.
that grows together with another bacterium, but it's not a mycolata. So it's probably in your gut, regulating some other bacteria.
So everyone does have a bit of Jason in their gut.
Yeah.
Love it. They are, yes.
So you think these parasitic bacteria are acting together like a white blood cell or an environmental immune system?
So are these, I guess, bacteriophages and stuff acting as like an environmental immune system for the wastewater treatment plants?
They could be. I don't think they're maybe acting like, but they're definitely controlling. I think that they do control the bacterial populations in the wastewater treatment plant. Yeah.
Yes, we're quick, yeah.
Are these found something that occurred, say, in the last 20 or 30 years?
So have they always been there, or have they...
The foams? Yeah. Always. They've been, so it's been more than 30 years. So there's been foaming events. So the very first report in the literature of a foaming event in wastewater treatment plants was published in the 1960s. So, and that was the very first report, and that's when people started to study what organisms are there, and then it took them about several years to work out these hydrophobic bacteria.
So you have to go.
Yes.
All right, we might take one last very quick question up the back there.
It's not immediate. It's going to be a while.
How can we submit Jason as an ADHD icon?
I think everyone heard that anyway, but how are we making Jason an ADHD icon is Matt's question up the back there.
I have no idea. I need everybody's help.
Maybe we could ask Jason. Jason, any comments up the back there? He's probably asleep. I don't know.
Yeah. No, he's awake.
Oh, it's night time. He's awake. All right, perfect. We might give Steve a round of applause.